A very good morning to one and all. Today we are going to learn about electron transport chain. So electron transport chain, this is the session. Already I have explained you in your previous sessions about mitochondria, structure of mitochondria, isn't it? So this is the structure of mitochondria, and these are the different complexes that are utilized during in electron transport chain, and this is the overall view of electron transport system. Got it? So today we will be learning about, today's session is about electron transport chain. From the name itself it indicates that here from one substance to another substance, electrons are being transferred from one substance to another substance. So in a form of a chain model, isn't it? So from the name itself it indicates that electron transport chain, and this electron transport chain is linked with your mitochondria, means it is taking place in animals, isn't it? So if at all electron transport chain is taking place in bacteria, then it will have this particular chain form of reaction will take place in your plasma membrane, because in bacteria there is no mitochondria is present, right? So coming to electron transport chain in animals, so before going into the electron transport chain, what is the mechanism lying behind your electron transport chain? You should be aware of the structure of mitochondria, as mitochondria is known as the powerhouse of the cell, isn't it? So in the previous session only I have explained you the structure of mitochondria and why ATP is termed as a high energy compound, and it is the currency of the cell, isn't it? And mitochondria is termed as powerhouse of the cell. So all these all these questions and all the why, what, when, how it happened, all these things you will be learning in this session. So this is the structure of mitochondria. So coming to the structure of mitochondria, this is your outer membrane. So this is your outer membrane. Okay, this is your intermembranous space, and this is your inner membrane. In between your outer membrane and in between your inner membrane, intermembranous space is present, and this is your matrix. Khali space in your mitochondria is matrix, and these incursions are cristae, and these are F0, F1 particles. These are phosphorylating particles where your ATP molecule is synthesized. So this is the overall ultrastructure of mitochondria. Got it? So all your electron transport chain system, the assembly of electron transport chain, all these complexes are present over on your this thing. What do you say? Cristae, near your cristae. In the open matrix, in your matrix, the cristae is present, isn't it? So here, you are in between your two cristae, two incursions, your your electron transport chain system complexes are present. So this is complex 1, complex 2, complex 3, 4, and this is 5. Okay. So these are the different complexes that are involved in your electron transport chain. So there are different complexes, there are different substances which are utilized for your electrons to begin transfer from one substance to another substance, like your cytochromes. These are the different substances that are used for your electron transport chain, isn't it? Cytochromes, iron, sulfur, cluster, protein substances, coenzyme Q, and several FMN substances, flavonomononucleotide, and NAD. Isn't NADH? So these are the different substances that are used for your electrons to be get transferred from one substance to another substance. So before going into your electron transport chain mechanism, there is a thing that you should be learn that about your glycolysis and your Krebs cycle, isn't it? So in your glycolysis from the glucose converter, so these are the different substances, right? So in glycolysis, glucose is being converted into pyruvic acid, isn't it? So here one NADH molecule is reduced to release one H plus ion. So this is a 6-step process where before entering into your electron transport chain, there is a 6-step process where hydrogen atoms are released. Hydrogen atoms are released and they will be utilized during your electron transport chain. So here one reduced NAD molecules are released. Next from pyruvic acid, acetyl CoA molecule is released. Here one more NADH molecule is released and with a H plus ion, and this acetyl CoA molecule will enter into your TCA cycle, isn't it? So here 4 NADH molecules are released. So totally 1, 2, 3, 4, 5, 6. Totally it is a 6-step process where 6 H plus ion molecules are released and these protons will go and interact with your electron transport chain assembly in order to transfer your electrons from one substance to another substance, and ultimately there is a release of ATP molecule, which is nothing but your high energy compound, and this energy is utilized by your cells in your every part of your body, and that energy is utilized by the human body in your daily routine work, isn't it? So this is the actual story before your electron transport chain mechanism. So how this, before going to this mechanism of electron transport chain, before your mitochondrial structure, before your complexes involved in electron transport chain, so there is a flow, there is a 6-step process where from glucose to pyruvic acid there is a release of one NADH molecule and H plus, and this here one H plus ion is released and here one more H plus. So this is a 6-step process where 6 H plus ions are released. So each pair of hydrogen ion will release 3 ATP molecules. So that is the summary of electron transport chain. So this is how, this is a 6-step process. So after finishing this 6-step process, you will be learning about the structure, ultrastructure of mitochondria. See, your glycolysis will take place in cytoplasm, isn't it? So here glycolysis will take place, and in your matrix Krebs cycle or TCA cycle will take place, and in your in your inner membrane ETC is taking place, isn't it? So all these 3 steps, all these 3 mechanisms are interlinked for the production of your energy. Ultimately, energy is produced in your 5th complex. This is nothing but ATP synthase complex, right? So this is the order of your synthesis of your energy in mitochondria in animals. So initially in cytoplasm, assume that this is a cell. In your cytoplasm, glycolysis will take place, and after in your glycolysis, glucose, the end product of your glycolysis is pyruvic acid, where one NADH molecule and H plus ion is released. So after this pyruvic acid will enter into your TCA, that is your Krebs cycle, and to form your acetyl CoA. This acetyl CoA will enter into your Krebs cycle, that is your TCA cycle, to form 4 NADPH molecule, NADPH and H plus ions, isn't it? So totally 6 NADPH and H plus ions are released. So here in your inner membrane, your ETC assembly is organized, and here in the 5th complex your ATP is synthesized, that is your high energy compound. So these are the different complexes that are involved, that are utilized during your electron transport from one complex to another complex. Complex 1, that is NADH dehydrogenase. Complex 2, that is succinate coenzyme Q reductase. Complex 3, coenzyme Q, cytochrome C reductase. This is cytochrome C. This is complex number 4. And complex number 3. Coming to complex number 4, cytochrome oxidase. And complex 5, ATP synthase. Here your actual oxidative phosphorylation will take place, and with the electrons or with the ATP that is released up to your complex 4, whatever electrons are released, those electrons will come in contact, electrons will come in contact with this complex 5, and here actual ATP synthesis will take place. That is in your F0, F1 pattern. These are phosphorylating subunits. Up to here, these are the 4 subunits, 1, 2, 3 and 4. Here electrons are being transferred from one complex to another complex, from one substance to another substance. There are different substances are there, different complexes are. These are the complexes, and these complexes include different substances utilized by your electrons in order to be easily transferred. So those substances, once again I am repeating, those substances are cytochromes and coenzyme Q, iron sulfur cluster proteins, and your NADPH molecule, isn't it? So those are the different and many more proteins and ions like magnesium. All those are present for your easy transfer of electrons from one complex to another complex. So as I said earlier, all these energy containing complexes, your electron transport chain assembly is present in your inner membrane of your mitochondria. So this is your inner membrane. This is your intermembranous space. This is your outer membrane, isn't it? So this is your outer membrane. See, this is your outer membrane. Isn't it? This is your intermembranous space. Okay, intermembranous space. This is your inner membrane of your mitochondria. Got it? So this is your outer membrane of your mitochondria, and this is your intermembranous space, and this is your inner membrane. See, look here, this is your outer membrane, this is your intermembranous space, and this is your inner membrane. All your electron transport chain 4 complexes are present, and your phosphorylating subunit, that is your complex 5, where your ATP is synthesized, where your energy is synthesized. These are the 5 complexes. So this is complex 1, 2, 3, 4 and 5. Here at last this is your here your ATP molecule is synthesized, that is your oxidative phosphorylation, that you will be learning in the next session. Up to here, how your electrons are being transferred from one complex to another substance. What is the mechanism lying behind the transfer of electrons from complex 1 to complex 4, that you will be learning in this session. In the next session, you will be learning the ATP synthase and the synthesis of your ATP molecule with the help of ATP synthase, that is your complex 5, that is nothing but oxidative phosphorylation. Isn't it? So this is the next session. Up to here, today in this session you will be learning about different mechanism that is used, present here for the electrons to get transferred from one complex to another, from one substance to another substance. See, already you have learned that what is the before process beginning of your electron transport chain, before the mechanism of electron transport chain starts. This is a 6-step process. That 6-step process is nothing but, see, once again I am repeating, that is a 6-step process where in glycolysis, glucose, the end product of glycolysis is pyruvic acid. From this will take place in your cytoplasm. See, this is your cytoplasm. So this pyruvic acid will convert into acetyl-CoA. Then here one more NADPH and H plus ion are released, and this here one more NADPH and H plus ion released. So this is a second step. And from acetyl-CoA, this acetyl-CoA will enter into your TCA cycle, that is in your matrix. Here 4 steps your NADPH molecule plus H plus ions are released, isn't it? So all these 3 H plus ions All these three will enter into your electron transport chain assembly, that is your inner membrane of your mitochondria. So that is nothing but your, this is your inner membrane. So this, how this inner membrane is organized, or how these electrons are being transferred from one complex to another complex, that you will be learning in your 4 complexes. And in the 5th complex, the transferred electrons, because of the transfer of these electrons, your ATP molecule is synthesized with the help of the enzyme ATP synthase, and that particular step is nothing but your oxidative phosphorylation. So this totally electron transport chain includes your first 4 complexes, and your oxidative phosphorylation includes your last 5th complex where your ATP molecule is synthesized, isn't it? So now coming to the overview of electron transport system, how your electrons are being transferred from one substance to another substance, that you will be learning in your this mechanism. See, before going to the mechanism, what do you mean by oxidation? Oxidation means nothing but loss of electrons. And what do you mean by reduction? Reduction is nothing but gain of electrons. So electron transport chain is nothing but the series of reaction which includes oxidation and reduction steps. So that is nothing but electron transport chain. The mechanism lying behind your electron transport chain is, it is a series of both oxidation and reduction steps. So with the help of these two terms only, with these two action of oxidation and action of reduction, with this particular two methodology only, your electrons are being transferred from one complex to another complex. So this, the mechanism lying behind this transfer of electrons from one complex to another, that you will be learning here. So this is your complex one. This is your NADPH molecule which that has been released, and this NADPH molecule will be get reduced to into NADH. So during its reduction, then what happens? 2 electrons and 2 proton molecules are released. So the released electrons are accepted by FMN. FN is tribonucleotide, mononucleotide. So after accepting, so means NADH is donating FMN 2 electrons, then what happens? This will be get oxidized. So the first initially it is reduced form. So after donating, after loss of 2 electrons, so initially, see once again I am telling, this is initially reduced form, this complex, isn't it? So this reduced form of complex, by donating 2 electrons to FMN, so after reduction what happens? It will produce 2 electrons and 2 protons. So 2 protons are released in the space, intermembranous space, and the 2 electrons that are released will be accepted. So this NADH will donate 2 electrons, it will lose 2 electrons to donate electrons to FMN. So this is known as, this is converted into oxidized form, and this is converted into reduced form, isn't it? So initially it is reduced form. After donating, after losing 2 electrons and 2 protons, these 2 electrons are accepted by FMN. So this will be converted to reduced form, and this will be converted into oxidized form. So initially in the before beginning of your class only I have explained, I have said you definition of oxidation and reduction. Oxidation means loss of electrons, leaving of electrons. Here loss of electrons means it is donating electrons to FMN. So this is a acceptor and this is a donor. So this is oxidized form and this is reduced form. Again FMN will donate 2 electrons to cytochrome B, hence it is converted into oxidized form, and this cytochrome B will be converted into reduced form. Again from this complex cytochrome B, again it will donate 2 electrons to cytochrome C complex, and it will be converted into reduced form, and this will be converted into oxidized form. Oxidized form to reduced form, how it is being converted? See, the mechanism is nothing but oxidation means losing of electrons, giving up of electrons, donating electrons to another substance. So this particular substance which is donating or losing or giving up of electrons, this particular substance is known as oxidized form. So these 2 electrons are accepted by the another substance that is outside to which it is giving. So this is termed as reduced form. So NADH is a reduced form initially. So this NADH will donate or will give up 2 electrons and 2 protons. So these 2 electrons are accepted by FMN. So this is known as oxidized form, and FMN is known as reduced form because it is accepting 2 electrons, isn't it? Next, FMN again is giving up 2 electrons that it has accepted, right? So again it is giving, it is losing, it is donating 2 electrons to cytochrome B. So this is converted into reduced form, and again this is converted into oxidized form. So next, this reduced form will convert it into oxidized form by giving 2 electrons to cytochrome. So the same process is taking up to your cytochrome A3. So reduce to oxidize, reduce to oxidize, reduce to oxidize. Reduce, initially it will be in reduced form because it is accepting electrons. Next, when it is giving up electrons to the next substance, it will convert it into oxidized form. Reduce to oxidize, reduce to oxidize, reduce to oxidize. Next, in the last, so in every first, third and then the fifth step, what happens? First step and then the second step and then the fourth step complexes, one ATP molecule is released. So this ATP, how it is released? During this reduction, what happened? ATPs will by production by hydrogenation, ATP molecule is produced. So these 3 ATP molecules will club up with your 5th ATP synthase enzyme to produce energy. So that is in the next step. So up to here, these complexes are changed into reduced to oxidized form. Reduced to oxidize, reduced to oxidize, reduced to oxidize. Why reduce? Because it is giving up electrons. It is losing electrons. Sorry, it is gaining electrons. That is reduced. To oxidize means it is accept. Okay, so this is in a chain form. So oxidize, first it is reduced form. After reduction, it is donating 2 electrons. It has released 2 electrons and 2 protons. So the released electrons are accepted by FMN. So this is a donor to FMN. So this is oxide form. So reduce to oxide form. Reduce to oxidize. Reduce to oxidize. So see. So this is a stepwise series of reduced to oxidation steps. So this is known as redox reactions. Redox reactions. Isn't it? So oxidation means loss of electrons, and reduction means gain of electrons, isn't it? So this is a 6-step process. 1, 2, 3, 4, 5 and 6. So after this is the steps. Okay. So here totally in your intermembranous space or in your inner membrane, all the substances all the complexes that are involved in your electron transfer the mechanism lying behind it's for your electron transfer is nothing but redox reaction. Oxidation means loss of electron and reduction means gain of electron. Here, see once again I am repeating, NADH plus will give, it is initially in a reduced form, isn't it? When it is released from here, isn't during glycolysis, during your production of acetyl-CoA, during your TCA cycle, all these NADH and H plus ions that are released are in reduced form. So the reduced form of NADH, single NADH molecule will come in contact with your electron transport chain. So the one compound that has first interacted with your electron transport chain, so this is initially in reduced form. So after giving up 2 protons and 2 electrons, then what happens? The electrons that it has released will be accepted by FMN. So it has released 2 electrons, so the reduced form will be changed into oxidized form. Now this is reduced form. Why? Because it has gained 2 electrons, isn't it, from NADH. Now the gain 2 electrons from NADH in the FMN, this FMN will also lose, will also give up 2 electrons, will also donate 2 electrons to cytochrome B complex. Now this cytochrome B complex will be converted into reduced form, and this is converted into oxidized form. Next, this cytochrome B complex will again release 2 electrons to cytochrome C. So this is losing electrons, so it is oxidized form, and this is reduced form because it is gaining 2 electrons. Next, cytochrome C will lose 2 electrons, so it is oxidized form, it is converted into oxidized form, and cytochrome A is converted into reduced form. Now again cytochrome A will release 2 electrons, will lose 2 electrons to cytochrome A3, and hence this is converted into oxidized form, and this is reduced form, and this is oxidized form. Now the forms, this reduced form of cytochrome A3, which is having 2 electrons, this electrons should be accepted by an oxygen. So there is an absence of oxygen. So by involving oxygen, here hydrogen H2O molecule is released from these 2 protons that are released during your initial step of your electron transport chain. So these 2 H plus ions, protons, will come and interact with your last form to release electrons in your cytochrome A3, and with the involvement of oxygen. So this last step here, with the involvement of oxygen, this is known as oxidative phosphorylation, that is where your ATP molecule is inserted. So this is a different mechanism that you will be learning in your next session, and here up to here your water molecule and oxygen, by the reduction of your electrons and with the combination of 2 proton molecules, water molecule is released, and hence there is a formation of ATP molecules. So how this ATP molecule is generated, that you will be learning in your 5th complex, that is oxidative phosphorylation. So this is nothing but the overall view of electron transport chain. Okay, this is the overall mechanism of electron transport chain. So this is nothing but electron transport chain. See, once again I am repeating, electron transport chain, before entering into the mechanism of electron transport chain, it is a 6-step process. In this 6-step, glycolysis, first initially you will consider glucose. In glycolysis, glucose is converted into pyruvic acid, where 1 NADPH molecule and H plus ion are released. So that take place in your cytoplasm. This is glycolysis. Next, the formed pyruvic acid will convert it into acetyl-CoA by release of 1 more NADH and H plus, and this acetyl-CoA will enter into your matrix, that is your Krebs cycle or TCA, tricarboxylic acid. Okay, this is your TCA. So from here to here, this is a 6-step process where in glycolysis 1 NADPH and H plus ion are released, and from pyruvic acid to acetyl-CoA 1 NADPH and H plus ions are released. That is the second step. And in your TCA cycle, that is your Krebs cycle, in your matrix 4 NADPH and H plus ions are released. So after the release of this NADPH and H plus ions, electron transport chain, this NADPH and H plus will enter into your electron transport chain, that is your inner space of your mitochondria. So this is a outer membrane space, this is your intermembranous space, this is your inner membrane. So here all the 5 complexes of electron transport chain are present. So this is complex 1, NADH dehydrogenase, complex 2, succinate coenzyme Q reductase, complex 3, coenzyme Q cytochrome reductase, complex 4, cytochrome oxidase, complex 5, that is ATP synthase. Here your actual ATP molecule is synthesized with the phosphorylating subunits, that is F0 and F1. This particular session, complex 5, you will be learning in your next class. So up to your 4 complexes, complex 1 contains 46 proteins, complex 2 contains 5 proteins, complex 3 contains 11 proteins, and complex 4 contains 13 proteins. So this NADH, before going into the mechanism, this inner membrane, all the 4 complexes are run by the mechanism of redox reactions. Redox reactions means both in a combination of oxidation and reduction. Reduction and oxidation. Oxidation means loss of electrons or giving up of electrons, donating electrons. Reduction means gain of electrons, accepting electrons. So here how redox reactions are taking place. This NADH plus is already in a reduced form. So this reduced form will give up 2 protons and 2 electrons, and these 2 protons are taken up by FML. So this FMN is reduced form, and this NADH is converted into oxidized form. Next, FMN will again give up 2 electrons to cytochrome B complex. So this is oxidized form and this is reduced form. Again, cytochrome B is converted, will give up 2 electrons to cytochrome C complex. So this is oxidized form and this is reduced form. Next, cytochrome C will give up 2 electrons and this is converted into oxidized form and this is into reduced form. Again, the cytochrome A will give up 2 electrons to cytochrome A3 complex. So this is oxidized form. This will give up 2 electrons and this will give up 2 electrons. So this cytochrome A3 is known as reduced form, isn't it? Now, without there is no involvement of oxygen. So when oxygen is involved, that is the oxidative phosphorylation. When oxygen is involved, the release of 2 hydrogen ions, that is your protons, will go and couple up with your 2 electrons that are released during your cytochrome A3, in your cytochrome A3 substance, near your cytochrome A3 substance, and here one water molecule is released. So this is nothing but your overall view of electron transport chain. Overall chain of electrons, how are being transferred from one substance to another substance, that is nothing but your electron transport chain. Different components, or these are different components. Each complex contains different components. Each complex, that is complex 1, 2, 3, 4, 5, contains different components that are utilized by the mitochondria, by these complexes to transfer your electrons. Those complexes are nothing but your cytochromes, iron sulfur cluster proteins, and the coenzyme Q, succinate dehydrogenase, and NADH dehydrogenase. That's all. Any doubts? So just pin me, I will clear your doubts. Thank you.